خلينا ندرس examples of small molecule neurotransmitters. راح نقضي وقت كويس على هدول الثلاث لأنه they are well studied. We have a wealth of information regarding هدول small molecule neurotransmitters. Well, regulation applies to other neurotransmitters as well, small molecule neurotransmitters. So, هدول the synthesis of these neurotransmitters اللي هم dopamine, norepinephrine, and epinephrine, their synthesis requires a number of cofactors اللي هم S-adenosyl methionine. فعندنا methyl transfer. طيب، وهذا الميثيل transfer requires folate وفيتامين B12, so keep that in mind. Pyridoxal phosphate is needed as well, or vitamin B6 for transamination with decarboxylation. Tetrahydrobiopterin is another cofactor, another organic cofactor. Tetrahydrobiopterin يمكن ما سمعتوا فيه قبل هيك، لكن it is synthesized from GTP and it exists as dihydrobiopterin or tetrahydrobiopterin. Do not memorize this synthetic pathway, by the way. This is the structure of the tetrahydrobiopterin. Synthesis of these three neurotransmitters takes place in the liver, or we can get it from diet, and we can synthesize it from phenylalanine by phenylalanine hydroxylase. فبصير في hydroxylation of this tyrosine, and then tyrosine can enter the neurons by active transport. Now, phenylalanine is an essential amino acid. We have to get it from diet, but once we do, we can synthesize tyrosine. عشان هيك tyrosine is known as a conditional essential amino acid, أو conditional non-essential amino acid, لأنه we get it from an essential amino acid, but once we get the essential amino acid, that is phenylalanine, we can synthesize tyrosine. So that's reaction number one. Then tyrosine is hydroxylated again by tyrosine hydroxylase into an intermediate known as DOPA, and this is a rate limiting reaction because tyrosine hydroxylase is highly regulated, so it's a slow reaction, and it requires tetrahydrobiopterin. Tetrahydrobiopterin is needed for hydroxylating rings. Okay, so عشان هيك احنا بدنا tetrahydrobiopterin. ما علينا. فعندنا هذا الانترميديت اللي هو دوبا. هذه الرينج اسمها كاتيكول. عشان هيك هدول الثلاث نيورو ترانسميترز يعرفوا باسم كاتيكول امينز. كاتيكول امينز. ما علينا. فعندنا هذه الدوبا. هذا الرياكشن بيصير بالسايتوسول. برضه بالسايتوسول بيصير عندنا الرياكشن اللي هو decarboxylation of دوبا عشان نعمل دوبامين. وهذا الرياكشن decarboxylation it requires vitamin B6 أو pyridoxal phosphate. What happens then إنه الدوبامين هذا بيصير له packaging inside vesicles. okay, وبالvesicles بتحول إلى norepinephrine. والnorepinephrine بعدين بيطلع لبرا الvesicles and it gets converted by methyltransferase enzyme into epinephrine اللي هو عندنا الميثيل جروب هاي اللي موجودة عندنا هون. طيب، فعندنا hydroxylation فحطينا hydroxyl جروب هون، بعدين عندنا decarboxylation شلنا هاي الجروب، بعدين عملنا another hydroxylation reaction اوكي حطينا الهيدروكسيل جروب right here، وبعدين عملنا methyl transferase methyl transfer reaction حطينا methyl group right here. هذا هو الابينفرين. طيب ناخذهم واحد واحد. هلا ناخذهم واحد واحد. So tyrosine بيصير له conversion لدوبامين via dopa. Dopamine is then packaged بصير الرياكشنز هذه بالسيل periphery. بصير packaging للدوبامين inside vesicles, and these vesicles wait for the influx of calcium ions, and they fuse with the plasma membrane, releasing dopamine outside. Dopamine binds to a receptor على postsynaptic nerve, and then it has to be inactivated, and it gets inactivated by one, reuptake. Most of it would be taken up again by the presynaptic neuron, and inside cells it can be repackaged or it can be inactivated by monoamine oxidase in the mitochondria. Some of the dopamine would leak out into the bloodstream وبيصير transfer or transport into the liver, where it gets inactivated by also these two enzymes أنا بنحكي عنهم, or it can be taken up by neighboring cells including the postsynaptic neuron, where it can also be inactivated. In terms of the re-entry of dopamine inside the presynaptic nerve, it takes place via a transporter, sodium ion dependent transporter, and this is the target of cocaine and amphetamine, leaving dopamine in the synaptic cleft for a longer time so that it can induce excitation, energy, and the person would be like fully aware and excited, in other words. هذا بالنسبة للدوبامين. norepinephrine بصير في عنا packaging of dopamine inside vesicles, وهاي الفيسيكلز بيصير فيها conversion of dopamine ل norepinephrine. طيب وبعدين again calcium ion influx, fusion of vesicles with the plasma membrane, release of norepinephrine into the synaptic cleft, ونفس ال process of inactivation أو termination of signaling اللي هو reuptake, uptake by neighboring cells, diffusion, enzymatic inactivation, and so on. أما بالنسبة للإبنفرين إنه في عنا النورابinephrine is now inside vesicles. It leaks out of these vesicles and it gets converted للإبنفرين. And then epinephrine is packaged again inside vesicles. ونفس الحكي fusion, release, reuptake, uptake by neighboring cells, diffusion into and transport to the liver, enzyme inactivation, and so on. هلا بالنسبة للpackaging of these neurotransmitters أو the catecholamines inside vesicles, خصوصا بنحكي على الدوبامين والابنفرين, these are transported inside vesicles via an ATP dependent process that requires also the efflux of protons from the vesicles themselves. فاللي بيصير عندنا actually إنه we have a transporter, a vesicular ATPase transporter that consumes ATP for the entry of protons inside vesicles. So the protons, the vesicles are acidic inside, low pH, and there are a lot of positive charges inside cells versus negative charges outside in the cytosol. فبصير في عنا exchange transport as well between the protons and the monoamine neurotransmitters. And this transporter right here, the VMAT, the vesicular monoamine transporter, can be targeted by certain inhibitors or drugs, leaving the monoamine neurotransmitters in the cytosol for a longer time, where they can be inactivated enzymatically, and that causes depletion of these neurotransmitters. نتكلم على inactivation of these neurotransmitters. So it happens enzymatically. We're talking about the enzymatic inactivation. It happens via two enzymes. The first one is known as monoamine oxidase. The other is known as the catechol or methyltransferase. It doesn't matter which one starts first. يعني ممكن monoamine oxidase وبعدين المethyltransferase أو ممكن methyltransferase بعدين monoamine oxidase. Since we're talking about a methyl transfer reaction, فعشان هيك احنا بحاجة لفيتامين B12 and the folate عشان for the synthesis of the S-adenosylmethionine inside cells. طيب الاشي الثاني as well إنه these enzymes exist in the presynaptic neuron as well as adjacent cells مثل الglial cells والpostsynaptic neurons as well. وعندنا drugs حكينا هذه بالprevious slide إنه drugs that result in inhibition of the transfer or the transport of these neurotransmitters inside vesicles causes depletion of these neurotransmitters لأنه هدول الانزيمز would act on these neurotransmitters, degrading them, inactivating them. Something else إنه بالParkinson's disease لما يكون عندنا high level of dopamine that results in the presence of homovenilic acid اللي هو end product بالurine. فهدول drugs would result in the inactivation of the dopamine with higher level of homovenilic acid in the urine. But people with Parkinson's disease, they have reduced level of the homovenilic acid in urine. وبالنسبة للregulation حكينا إنه أول reaction اللي هو tyrosine that is catalyzed by tyrosine hydroxylase هو rate limiting reaction لأنه هذا الإنزيم is regulated and it is regulated short term and long term. Okay, فعندنا short term regulation and long term regulation. Long term regulation على طول we're talking about gene expression, we're talking about transcription with translation and it takes time, takes hours. فلما يكون عندنا prolonged sympathetic neuronal activity لما يكون عندنا a lot of firing بالneurons حيصير في increased transcription of tyrosine hydroxylase as well as dopamine hydroxylase اللي بيحول dopamine إلى norepinephrine. short term regulation بصير at the level of enzyme activity. فلما يكون عندنا high level of cytosolic catecholamines they will compete with the tetrahydrobiopterin binding للإنزيم فبصير في عندنا inhibition of the enzyme. In addition ممكن يصير في activation by depolarization. فلما يكون عندنا a lot of depolarization حيصير في increased activity لبروتين كاينيز C, بروتين كاينيز A, calcium calmodulin dependent kinases اللي بيعملوا dephosphorylation اللي بيعملوا phosphorylation للتيروسين hydroxylase and as a result activation لأنه بصير في عنا increased binding increased affinity between tetrahydrobiopterin and the enzyme.